Okay, Ra? Voice clear, Ra? So clear. So clear. Voice is clear.
Yes, sir.
Okay. Today our topic is so today so our topic is cross-relation. So cross-relation acid metabolism. So cross-relation acid metabolism. So are commonly known as CAM. So today our topic is cross-relation acid metabolism. So cross-relation acid metabolism. So what is the cross-relation acid metabolism? The cross-relation acid metabolism is also known as dark reaction. So it is also known as dark reaction. The cross-relation acid metabolism is also known as dark reaction. Sorry, dark dark carbon dioxide fixation. So cross-relation acid metabolism is also known as dark carbon dioxide fixation. So what is the cross-relation acid metabolism? So what is the cross-relation acid metabolism? So what is the cross-relation acid metabolism? In some plants, in some some non-halophytic non-halophytic in some non-halophytic succulents, in some non-halophytic succulents and semi succulents, in some non-halophytic succulents and semi succulents, the acidity, so during the night time, night time acidity, so acidity is increases in some non-halophytic succulents and semi succulents. During the night time acidity is increased. acidity is increased. During the daytime it is decreased. So during the night time it is increases and during the night time it is decreases. So it was first observed. So it is first observed in Crocylasian. So Crocylasian plant. So Crocylasian plant. So Crocylasian plant. Bryophyllum. So bryophyllum. So bryophyllum calcinum. Bryophyllum calcinum. Bryophyllum calcinum. Hence it is called. So hence it is called crossylation acid metabolism. So crossylation acid metabolism. So clear. In some non-halophytic saccharides and semi-succulent plants, during the night time, acidity is increases. During the daytime, during the daytime, acidity is decreases. Acidity is decreases. So it was first observed. So it was first found. It is found. It was first found. It was first found in a Crocylasian plant, Bryophyllum, Bryophyllum calcinum, Bryophyllum calcinum. Hence it is called Crocylasian. Hence it is called Crocylasian acid metabolism. So it is clear. The crossylation acid metabolism is also known as dark carbon dioxide fixation under natural conditions in some non-halophytic succulents and semi-succulents. During the night time, the acidity is increases. During the daytime, it is decreases. So it was first found in Crocylasian plant Bryophyllum calcinum. Hence it is called Crocylasian acid metabolism. So Crocylasian acid metabolism. The term CAM, Crocylasian metabolism, and the plants are known as CAM plants. The plants are known as CAM plants. The term CAM is coined by the term the term CAM is coined by the term CAM is coined by Ronson. The term CAM is coined by Ronson and Thomas. Coined by Ronson and Thomas. So is it clear? So the plants are called CAM plants. The term CAM is coined by Ronson and Thomas. So in which plant the CAM cycle is occurred? In which plants the CAM cycle is occurred? So it is generally occurred in bryophyllum. So bryophyllum and Colonchi, sedum, clinia, crassula, and opuntia. So clear. So generally the CAM cycle, they are examples of CAM plants. The examples of plant, Bryophyllum, Colonchi, sedum, clinia, crassula, and opuntia. So it is also found in the families. So it is also found in some families like Crassulaceae, Coctaceae, Orchidaceae, Portucaceae, Agavaceae, Opuntiaceae, etc. So clear. The camp cycle is also found in some families like Crossuliaceae, Coctaceae, Orchidaceae, Portucaceae, Opuntiaceae, Againstvaceae, etc. So clear. So first of all, so Crossuliacean acid metabolism. The Crossuliacean acid metabolism is also known as dark carbon dioxide fixation. So what is Crossuliacean acid metabolism? Under natural conditions, in some non-halophytic succulents and some semi-succulents, during the night time, SDT is increases. During the daytime, the SDT is decreases. It was first observed in Crossuliacean plant Bryophyllum colcinum. Hence, it is called Crossuliacean acid metabolism. And the plants are known as cam plants. The term cam is coined by, the term cam is coined by, the term cam is coined by Ronson and Thomas. So, in which plant the cam cycle is occurred? The plants are Bryophyllum, Kalanki, Sedum, Clinia, Crossula, Opuntia. So in some plants, so in some families like Crossuliaceae, the family members of Crossuliaceae, Coctaceae, Orchidaceae, Portucaceae, Agaveaceae and Opentiaceae. In these families, the plants show the camp cycle. So is it clear? So what is the speciality of camp plants? So what is the speciality of camp plants? So in camp plant, the stomata, in camp plant, the stomata remain closed. The stomata remain closed during the daytime. During the daytime, the stomata remain open or open during the night time. During the night time, to prevent transpiration, to prevent transpiration, to prevent transpiration in camp plant, the stomata remain closed during the daytime and open during the night time to prevent the transpiration. So the carbon dioxide fixation, the carbon fixation occurred during the night time. The carbon fixation occurred during the night time. Hence it is called dark carbon dioxide fixation. That's why it is called carbon dioxide, dark carbon dioxide fixation. So clear. So in camp plant, the stomata, the stomata remain closed during the daytime and open at night time to prevent the transpiration. So the carbon dioxide fixation occurred during the night time. So it is called dark carbon dioxide fixation. So clear. So what are the main reactions of cam cycle? The cam cycle generally occurred. The cam cycle generally occurred. It occurs two different different time intervals. Time intervals. So it occurs, the cam cycle occurs in two different time intervals. They are daytime and night time. The C4 cycle occurred in two different type of cells, but The CAM cycle occurred in two different times. They are daytime and next one is the night time. So during the daytime and during the night time. So what are the main reactions? So what are the main reactions in CAM cycle? So what are the main reactions in CAM cycles? So already I clearly mentioned the CAM cycle occurred. It occurred in two different time intervals. It occurred in two different time intervals. So they are daytime and next one is the night time. So during the daytime and next one is the night time. This is the night time. This is the daytime. So this is the night time and this is the daytime. So during the night time, the stomata is opened. During the night time, the stomata is opened. The carbon dioxide enter into the mesophyll cells. The carbon dioxide enter into the mesophyll cells and dissolved in water to form bicarbonate. This bicarbonate react with PEP. So PEP means phosphoenol pyruvic acid. This bicarbonate is react with phosphoenol pyruvic acid to form oxalo. So oxaloacetic acid. During the night time, the stomata is open. The carbon dioxide enters into the plant and dissolved in water to form bicarbonate. This bicarbonate reacts with phosphoenol pyruvic acid to form oxaloacetic acid. This oxaloacetic acid in presence of enzyme dehydrogenase. So it is converted into malic acid. So in presence of enzyme dehydrogenase, the enzyme name is dehydrogenase. Here the enzyme name is PEP carboxylase. Dehydrogenase to form malic acid. In this reaction, 1 mole of NADPH2 is reduced to form NADP plus. So malic acid is formed. This malic acid is formed during the night time. It is stored. So it is stored in vacuole. So it is stored. The malic acid formed during the night time. It is stored in vacuole. So clear. During the night time, the C4 cycle, the CAM cycle occurred in two different type of times. During the night time, the carbon dioxide, the stomata remain open. The carbon dioxide enter into the plant and dissolved in water to form carbonate. This bicarbonate react with phosphoenol pyruvic acid in presence of enzyme PEP carboxylase to form oxaloacetic acid. This oxaloacetic acid in presence of enzyme malic dehydrogenase to form malic acid. In this reaction, 1 mole of NADPH2 is reduced. So malic acid formed in this reaction during the night time, stored in vacuum. So during the daytime, the malic acid released from vacuole. So released from vacuole and it is decarboxylated. So released from vacuole and it is decarboxylated in presence of enzyme specific malic dehydrogenase dehydrogenase. So specific molecule dehydrogenase. So specific molecule dehydrogenase to form pyruvic acid. To form pyruvic acid. To form pyruvic acid. So in this reaction, one mole of NADP plus reduced to form NADPH2. NADP plus is converted into NADPH2. So clear. The carbon dioxide released from this reaction is accepted by RUBP and perform the C3 cycle. Thereafter, this pyruvic acid in presence of enzyme pepkinase or pyruvate kinase in presence of enzyme pyruvate kinase. So pyruvate kinase. So react with 2 moles of ATP. So ATP is converted to 2 moles of ADP. ADP to form phosphoenol pyruvic acid. Phosphoenol pyruvic acid. So these are the major reactions in the CAM cycle. So clear. So the CAM cycle occurred in 2 different time intervals. During the night time, the stomata is opened, the carbon dioxide enters into the plant and dissolved in water to form bicarbonate. This bicarbonate reacts with phosphoenol pyruvic acid in presence of enzyme pep carboxylase to form oxaloacetic acid. This oxaloacetic acid in presence of enzyme malic acid. So in presence of enzyme malic dehydrogenase converted into malic acid. The malic acid formed during the night time, it is stored in vacuole. So stored in vacuole during the daytime, it is released from vacuole and decarboxylated to form pyruvic acid and phosphoenol pyruvic acid. So these are the main steps in the CAM cycle. So these are the major events in the CAM cycle. So clear. So next one, the absorption of carbon dioxide. So during the daytime, during the night time, the malic acid during the night time, the carbon dioxide stored in the form of organic acid. So clear. So during the night time, the carbon dioxide is stored as organic acids. So organic acid like malic acids. This process is known as acidification. This process is known as acidification. So this is the one of the most important. So what is the acidification? In CAM plant, during the night time, the carbon dioxide stored as organic acids like malic acid. So this is called acidification. So during the daytime, the malate is decarboxylated. The malate or malic acid is decarboxylated. So the malic acid is decarboxylated and release the carbon dioxide. So this process is known as deacidification. So clear, there are two words, one is the acidification and second one is the deacidification. So acidification means the carbon dioxide stored as a organic acids is known as acidification during the night time. During the daytime, the malic acid is decarboxylated decarboxylated to form carbon dioxide. So carbon dioxide, this process is known as deacidification. So important one. One is the acidification and second one is the deacidification. So one is the acidification and second one is the deacidification. So clear. So in CAM plants, so one of the another important feature is RQ, respiratory quotient value. The respiratory quotient value of RQ plants is always 0. The, the RQ value of the RQ value of CAM plants are xerophytic plants. The RQ value is 0 because during the night time the oxygen is utilized continuously. So night time during the night time the carbon dioxide utilized continuously. So there is no evolution or carbon dioxide formation. So the RQ value is 0. So RQ value is 0 because of in CAM plant during the night time the oxygen is utilized continuously. There is no formation of carbon dioxide. So RQ value is 0. Generally RQ means, so RQ is equal to, so RQ means, so number of carbon dioxide. So carbon dioxide evolved. So number of carbon dioxide molecules evolved by number of oxygen molecules. So number of oxygen molecules are consumed. So here 0 by infinity. So it is always 0. So clear. So in CAM plants the RQ value is 0 or infinity because during the night time oxygen is utilized continuously for the synthesis of myelin. So no carbon dioxide is evolved. Hence the RQ value is 0. The RQ value is 0 in CAM plants. So clear. So this is the information about. The CAM cycle. So clear. So once again. So what is the CAM? So CAM cycle. So CAM cycle or cryoglycan acid metabolism. So cryoglycan acid metabolism is also known as dark carbon dioxide fixation. So what is the cryoglycan acid metabolism? Under natural conditions, in some non-halophytic succulents and semi-succulent plant, during the night time, acidity is increases. During the daytime, the acidity is decreases. This was first found in cryoglycan plant Bryophyllum calcinum. Hence, it is called cryoglycan acid metabolism, and the plants are known as CAM plants. The term CAM is coined by Ronson and Thomas. So examples of CAM plants: Bryophyllum, Kalanthi, Sedum, Clinia, Crossula, Opuntia. So some families like Crosslaceae, Coctaceae, Orchidaceae, Porticaceae, Agaveaceae, Opentiaceae. So they can fix the carbon dioxide in the form of cryoglycan acid metabolism. In CAM plants, the stomata remain closed during the daytime to prevent the transpiration. In CAM plants, the stomata remain closed during the daytime and open at night time. So the carbon dioxide fixation takes place during the night time. So it is called dark carbon dioxide fixation. The CAM cycle occurred in two different time intervals. They are daytime and night time, during the daytime and night time. So what are the important steps in the cramp cycle? So during the during the night time, the carbon dioxide enter the stomata is opened. The carbon dioxide enters into the plant and dissolved in water to form bicarbonate. This bicarbonate react with phosphoenol pyruvic acid in presence of enzyme PEP carboxylenes to form oxaloacetic acid. The oxaloacetic acid is converted into malic acid. This malic acid stored in vacuole during the night time. During the daytime, it is decarboxylated to form pyruvic acid and finally phosphoenol pyruvic acid. So phosphoenol pyruvic acid. In CAM plant, during the night time, the carbon dioxide stored as organic acid or malic acid. So it is called acidification. In CAM plant, during the daytime, the malic acid is decarboxylated. The malic acid is decarboxylated to form carbon dioxide, decarboxylated and release the carbon dioxide. This process is known as deacidification. This process is known as deacidification. So clear. The RQ value of CAM plants. So the RQ value of CAM plant is infinity. So mostly infinity. Or nearly zero, because of during the night time the oxygen is utilized continuously for the synthesis of malic acid. So there is no formation of carbon dioxide. Hence the RQ value is infinity or sometimes zero. So clear. This is the information about the CAM cycle. So clear. And next one. So another topic. So next one, another one. So next one is the photorespiration. So another important one, photorespiration. So another important topic is the photorespiration. So photorespiration. So what is the photorespiration? The photorespiration is also known as oxidative. So it is also known as oxidative photosynthetic photosynthetic so it is also known as oxidative photosynthetic carbon cycle. The photorespiration is also known as oxidative photosynthetic carbon cycle. The photorespiration is also known as oxidative photosynthetic carbon cycle. So it is also known as glycolate metabolism. Glycolate metabolism. So it is also known as glycolate metabolism because of the first stable product in the cycle is glycolate. The first stable product in this cycle is glycolate, hence it is called glycolate metabolism. So it is also known as C2 cycle. Because of the first stable product in the cycle is glycolate. The first stable product in the cycle is glycolate. So it is a two carbon compound. Hence Hence, this cycle is known as C2 cycle. Hence, this cycle is known as C2 cycle because of the first stable product in the cycle is glycolate. Hence, this cycle is known as C2 cycle. And next one, another important one. The scientist Hutch and Slack, they discover the C4 cycle. The Hutch and Slack, the photorespiration called as metabolic. They called as metabolic. So metabolic adject. So metabolic adject to Calvin cycle. Metabolic adject to Kelvin cycle. So the scientists Hutch and Slack called as photorespiration is a metabolic adject to Kelvin cycle. So clear. So photorespiration. So photorespiration, it is a process. So what is the photorespiration? So it is a process which involves loss of fixed carbon dioxide, loss of fixed, the loss of fixed carbon dioxide or the loss of carbon. So photorespiration is a process. So photorespiration is a process which involves loss of fixed carbon, loss of fixed carbon as carbon dioxide, as the loss of fixed carbon in the form of carbon dioxide in the presence of sunlight, in the presence of sunlight in chloroplast. In chloroplast is known as photorespiration. So photorespiration means it is a process. So it is a process which involves loss of fixed carbon, loss of fixed carbon in the form of carbon dioxide, in the form of carbon dioxide, in the form of carbon dioxide, in the form of carbon dioxide in presence of sunlight in chloroplast is known as photorespiration. So photorespiration. So clear. The loss of carbon takes place in the form of carbon dioxide. So it does not, the photorespiration, it does not produces, it does not. So it does not produces, it does not produces. So it does not produces any ATP and NADPH2. It does not produces any ATP and NADPH2. So it is a wasteful process. So it is a wasteful process. So photorespiration is a wasteful process. So photorespiration is also known as oxidative photosynthetic carbon cycle, are also known as glycolate metabolism because of the first stable product. The first stable product is glycolate. Hence it is called glycolate metabolism or glycolate cycle. So it is also known as C2 cycle because the first stable product is glycolate. It is two carbon compound, hence it is called C2 cycle. The photorespiration is called by Hutch and Slack. They called as metabolic adjunct to Kelvin cycle. So photorespiration is a metabolic adjunct to Kelvin cycle by Hutch and Slack. So what is the photorespiration? So it is a process, so in which the loss of fixed carbon in the form of carbon dioxide in presence of light in chloroplast is known as photorespiration. So it does not produces any ATP and NADPH2. It does not produces any ATP and NADPH2. So it is a wasteful process. So it is a wasteful process. So it occurs. So ఇది ఎప్పుడు జరుగుతుంది? It occurs usually when there is the high concentration of oxygen. It occurs it occurs only in presence of high high concentration high concentration high concentration of oxygen. The C2 cycle occurred only in presence of high concentration of oxygen. So it was first discovered by 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 Decker and Theo. The photorespiration was first discovered by Decker and Theo. Decker and Theo. The term photorespiration is proposed by the term photorespiration is coined by Krotkow. So Krotkow coined the term photorespiration. So photorespiration was first discovered by Decker and Theo, but the term is coined by Krotkow. So Krotkow coined the term. So Krotkow. So this is the R. So Krotkow coined the term photorespiration. Generally, where it is occurred, the photorespiration generally occurred in chlorophyllous tissue. So it is occurred only chlorophyllous. So only chlorophyllous tissue. The photorespiration generally occurred only in chlorophyllous tissue. So it occurs only in chlorophyllous tissue. So it is completely absent. So it is absent. The photorespiration is absent in C4 plants. The photorespiration is absent in C4 plants. So clear. So photorespiration. So what is the photorespiration? The loss of fixed carbon in the form of carbon dioxide in presence of light in chloroplast is known as photorespiration. It does not produces any ATP and NADPH2. So it is a wasteful process. So it is a wasteful process. So it is a wasteful process. So next one, it occurs only in presence of high concentration of oxygen. It occurs only in presence of high concentration of oxygen. It was first discovered by Decker and Theo. The term is coined by Krotkow. So generally it occurred only in chlorophyllous tissue. So it is completely absent. It is completely absent in C4 plants. So it occurs only chlorophyllous tissue. It is completely absent in C4 plants. So in which plant it is occurred? So it occurs only those plants. So it occurs మరి ఇది ఏ ప్లాంట్ లోపల జరుగుతుంది? It occurs only in those plants which have high carbon dioxide compensation point. The plants which have high carbon dioxide compensation. So compensation point. The photorespiration generally occurred only in those plants which have high carbon dioxide compensation point. So compensation point. Example C3 plants. Like tomato, wheat, oats, oats, green algae, green algae. So green algae like chlorella, etc. So clear. So what is the, so generally the photorespiration occurs only in those plants which have high concentration, high carbon dioxide component. So which plant the carbon dioxide compensation point is very high, those plant the photorespiration is occurred. The photorespiration occurs only in those plants which have high carbon dioxide compensation point. So clear. So what is the carbon dioxide compensation point? So what is the compensation point? The compensation point means the rate of photosynthesis, the rate of photosynthesis. So at a point where the rate of photosynthesis is equal to rate of photosynthesis, or rate of respiration. So rate of respiration. So rate of respiration. So rate of respiration is called compensation point. So is called compensation point. The rate of photosynthesis will be equal to the rate of respiration is called compensation point. So is called compensation point. So generally the photorespiration occurred only in the plants of, they consist of high carbon dioxide compensation point. So what is the compensation point? So compensation point means the point in which the rate of photosynthesis is equal to rate of respiration. So rate of respiration. So clear. So generally the photorespiration occurs only in the plants which have the high carbon dioxide compensation point. So compensation point. So what is the compensation point? So compensation point means the rate of photosynthesis. The rate of photosynthesis is equal to the rate of photosynthesis equal to rate of respiration. Rate of respiration at a point is known as compensation point. In this compensation, those have the compensation point is very high, carbon dioxide compensation point very high. In plants the photorespiration is occurred. So generally the carbon dioxide compensation rate is about generally in C3 plants. In C3 plant the carbon dioxide compensation point is 25 to 100 ppm. So 100 ppm or more than 50 ppm. In C3 plants the carbon dioxide compensation point is 25 to 100 ppm or more than 50 ppm. In C4 plants the carbon dioxide compensation point is less than, so is less than 10 ppm. So this is the one of the most important one. In CAM plant the carbon dioxide compensation point is 0 to 5 ppm. 0 to 5 ppm only. The photorespiration generally occurred in the plants. They consist of high carbon dioxide compensation point. So what is the carbon dioxide compensation point? The carbon dioxide compensation point means the rate of photosynthesis will be equal to the rate of respiration is called compensation point. The carbon dioxide compensation rate is about 25 to 100 ppm or more than 50 in C3 plants. It is about less than 10 ppm in C4 plants, and it is about 0 to 5 ppm 0 to 5 ppm in CAM plant. So clear. So next one, the photorespiration. The photorespiration occurs in three different cell organelles. Three different cell organelles. The photorespiration generally occurred in three different type of cell organelles. They are the photorespiration generally occurred in three types of cell organelles. They are chloroplast, so chloroplast and peroxisomes, chloroplast, peroxisomes and mitochondria. Mitochondria. The photorespiration generally involved in three different type of cell organelles. They are chloroplast, peroxisomes and mitochondria. So clear. So next one, the photorespiration. The photorespiration is mainly catalyzed by the enzyme Rubisco. The photorespiration is mainly catalyzed by the enzyme Rubisco. So Rubisco means ribulose 1,5, ribulose bisphosphate carboxylase and oxygenase. The photorespiration, the key enzyme for the photorespiration is the Rubisco. The key enzyme for photorespiration is Rubisco. The Rubisco act as carboxylase, so it acts as carboxylase at the same time oxygenase. So it acts as both carboxylase and oxygen. So when it is act as carboxylase, so when it is carboxylase, when the carbon dioxide concentration, when the carbon dioxide concentration is higher than oxygen concentration, it acts as carboxylase. When the carbon dioxide concentration is higher than oxygen concentration, it is act as carboxylase and perform the C3 cycle. When the oxygen concentration is higher than carbon dioxide concentration, it is act as oxygenase and perform the C2 cycle or photorespiration. So C2 cycle or photorespiration. So clear. When the oxygen concentration higher than the carbon dioxide, the Rubisco act as oxygenase. Now it is known as RUBP oxygenase. When it is act as oxygenase, is known as RUBP oxygenase. So RUBP oxygenase. So clear. The photorespiration is catalyzed by the only one of the key enzymes is the Rubisco. So one of the key enzymes, the Rubisco, it acts as both carboxylase and oxygenase. So when it is act as carboxylase and when it is oxygenase. When the carbon dioxide concentration is higher than oxygen, it acts as carboxylase and perform the C3 cycle. When the oxygen concentration is higher than carbon dioxide concentration, it is act as oxygenase. Now it is called RUBP oxygenase. So it performs the C2 cycle. So clear. The C2 cycle occurred in three different type of cell organelles. They are chloroplast, peroxisomes and mitochondria. The enzyme Rubisco play an important role in photorespiration. In C2 cycle, the Rubisco act as oxygenase. It is called RUBP oxygenase. So RUBP oxygenase. So what are the main reactions? In photorespiration? So the main reactions of photorespiration. So once we observe the main reactions of photorespiration. So generally The photorespiration occurred in three types of cell organelles. Three types of cell organelles. They are chloroplast. The first column is chloroplast, and second one is the peroxisomes. So first one is the chloroplast. So first one is the chloroplast, and second one. So second one is the peroxisomes. Peroxisomes is the chloroplast, and final one is the mitochondria. So final one is the mitochondria. The photorespiration occurred mainly three different cell organelles. They are chloroplast, peroxisomes, and mitochondria. The first reaction is occurred in chloroplast. So first reaction is occurred in chloroplast. This reaction is catalyzed by the enzyme. The key enzyme is the Rubisco RUBP. When the carbon dioxide concentration is lower than oxygen, the enzyme Rubisco acts as oxygenase. Now it is called RUBP oxygenase. So clear. So what happened in first reaction? So in first reaction, the RUBP. So in presence of high concentration of oxygen and low concentration of carbon dioxide, the RUBP react with oxygen. RUBP react with oxygen in presence of enzyme RUBP. So RUBP oxygenase. So in presence of enzyme RUBP oxygenase. So when the high concentration of oxygen, the RUBP react with, in presence of high concentration of oxygen, the RUBP react with oxygen in presence of enzyme RUBP oxygenase to form 3-phosphoglyceric acid. So 3-phosphoglyceric acid and 2-phosphoglycolic. So 2-phosphoglycolic acid. 2-phosphoglycolic acid. So clear. In first reaction, in presence of high concentration of oxygen, the RUBP react with oxygen in presence of enzyme RUBP oxygenase to form 2-phosphoglycolic acid and 3-phosphoglyceric acid. So clear. This 2-phosphoglycolic acid in presence of enzyme. So in presence of enzyme phosphatase. So in presence of enzyme phosphatase, it lose one phosphate molecule. To form glycolate. To form glycolate. So clear. So in presence of high concentration of oxygen, the RUBP react with oxygen to form 3-phosphoglyceric acid and 2-phosphoglycolic acid. This 2-phosphoglycolic acid in presence of phosphatase lose one phosphate molecule to form glycolate. So the glycolate formed in chloroplast, it is transferred to, it is migrate into peroxisomes. So glycolate formed in chloroplast migrate into peroxisomes. In peroxisomes, it is oxidized. So it is oxidized. In peroxisomes, it is oxidized. So it is oxidized in presence of enzyme. So in presence of enzyme, glycolic acid oxidase in presence of enzyme glycolic glycolic acid acid oxidase in presence of enzyme glycolic acid oxidase to form glyoxylate glyoxylate glyoxylate. So clear. So in presence of enzyme, the glycolate acid oxidase, the glycolate is oxidized to form glyoxylate plus hydrogen peroxide H2O2 hydrogen peroxide. The glycolate formed in chloroplast migrate into peroxisomes. In peroxisomes, it is oxidized in presence of enzyme glycolic acid oxidase to form glyoxylate and hydrogen peroxide. This hydrogen peroxide, this hydrogen peroxide in presence of enzyme catalase. So in presence of enzyme catalase, it split to form water and oxygen. So it split to form in presence of enzyme catalase, it split to form water and oxygen. So next one, the next product is glyoxylate. This glyoxylate, this is the one of the important reaction. This glyoxylate in presence of enzyme glutamate glyoxylate transferase. In presence of enzyme glutamate, so glutamate glyoxylate transferase. So enzyme name is glutamate glyoxylate transferase. So it react with ammonia. So it react with ammonia. Or glutamate. React with ammonia or glutamate. Glyoxylate, in presence of enzyme glutamate glyoxylate transferate react with, so participate in transamination reaction. This is the transamination reaction to form glycine. So glycine is one type of amino acid. So glycine is one type of amino acid. So clear, the glycolate formed in chloroplast transferred to peroxisomes. In peroxisomes, the glycolate is oxidized to form glyoxylate and hydrogen peroxide. This hydrogen peroxide in presence of enzyme catalase split to form water and oxygen. The glyoxylate so participate in transamination in presence of enzyme glutamate glyoxylate transferase to form glycine. So clear. The glycine formed in peroxisomes. The glycine formed in peroxisomes migrate into mitochondria. So migrate into mitochondria. So the glycine formed in peroxisomes transferred to mitochondria. In mitochondria, 2 moles of glycine. So this is the 1 mole, another mole. 2 moles of glycine. So 2 moles of glycine react to form. So 2 moles of 1 mole of glycine. So another mole of glycine. The glycine, 2 moles of glycine react. The glycine, 2 moles of glycine react to form. 2 moles of glycine react to form serine. So another amino acid is called serine. So clear. The glycine formed in peroxisomes migrate into the mitochondria. In mitochondria, 2 moles of glycine, so 2 moles of glycine react with each other to form another amino acid serine. So another amino acid serine. So in this reaction, 1 mole of carbon dioxide is released. At the same time, 1 mole of ammonia is also released. So 1 mole of ammonia is also released. So next one, oxygen is utilized. So oxygen, okay, nothing too important. So in this reaction in mitochondria, so 2 moles of glycine react with each other to form serine. In this reaction, so 1 mole of carbon dioxide and ammonia is released. So clear. So this is the post elimination burst of carbon dioxide. So ఇక్కడ కార్బన్ డైఆక్సైడ్ రిలీజ్ అవుతాను. సో ఇక్కడ ఆక్సిజన్ తీసుకోవడం జరుగుతుంది. జనరల్గా రెస్పిరేషన్ అంటే ఇంటేక్ ఆఫ్ ఆక్సిజన్ అండ్ అవుట్పుట్ ఆఫ్ కార్బన్ డైఆక్సైడ్. సో ఇక్కడ అదే జరుగుతుంది. ఇక్కడ ఆక్సిజన్ తీసుకున్నాం, ఇక్కడ కార్బన్ డైఆక్సైడ్ అనేది బయటికి రిలీజ్ అవుతుంది. సో అందుకే ద నేమ్ ఈజ్ కాయిన్డ్ బై ఫోటో రెస్పిరేషన్. సో రెస్పిరేషన్ మీన్స్ so absorption of oxygen and release of carbon dioxide, it is called respiration. సో ఇక్కడ ఆక్సిజన్ తీసుకుంటాను, ఇక్కడ కార్బన్ డైఆక్సైడ్ అనేది రిలీజ్ అవుతుంది. అందుకే దీనికి ఫోటో రెస్పిరేషన్ అని పేరు వచ్చింది. సో అట్లా మనకి ఏం ఫామ్ అయింది? సో సెరైన్ అనేది ఫామ్ అయింది. సో సెరైన్ ఫార్మ్డ్ ఇన్ మైటోకాండ్రియా, సారీ, ఇన్ presence of enzyme. The enzyme name is serine hydroxyl. Serine hydroxy, so hydroxy methyl transferase. So methyl transferase, the enzyme name is serine hydroxy methyl transferase. This reaction is catalyzed by the enzyme serine hydroxy methyl transferase. సో నోట్, ఇక్కడ ఇంపార్టెంట్ వన్ ఏంటిది అంటే, ఇన్ ఫోటో రెస్పిరేషన్ ద కార్బన్ డైఆక్సైడ్ రిలీజ్డ్ ఇన్ మైటోకాండ్రియా. The carbon dioxide released in photorespiration. The carbon dioxide released in mitochondria, not in peroxisomes and not in chloroplast. So next, the serine formed in mitochondria transferred to peroxisomes. In peroxisomes, it is transaminated. So trans means, so it is transaminated. So loss of ammonia, ఇక్కడ అమోనియా యాడ్ అవుతుంది, ఆ యాడ్ అయ్యే అమోనియా దీని నుంచి వచ్చింది. ఇట్ ఈస్ deaminated. So it is deaminated in presence of enzyme serine glyoxylate aminotransferase. Serine glyoxylate serine glyoxylate aminotransferase. So clear. The serine formed in mitochondria transferred to peroxisomes. In peroxisomes, it is deaminated in presence of enzyme serine glyoxylate aminotransferase to form hydroxypyruvate. So hydroxypyruvate. So clear. The hydroxypyruvate formed in peroxisomes, it is reduced. So it is reduced in presence of enzyme hydroxypyruvate reductase. In presence of enzyme hydroxypyruvate reductase, it is reduced to form glyceric acid. In this reaction, 1 mole of NADH2 reduced to form NAD+. So glyceric acid is formed. The glyceric acid formed in peroxisomes. So glyceric acid is formed in peroxisomes. It is transferred. The glyceric acid formed in peroxisome transferred to chloroplast. Chloroplast. In chloroplast, it is phosphorylated. In chloroplast, it is phosphorylated in presence of enzyme. So, in presence of enzyme kinase, it is phosphorylated to form 3-phosphoglyceric acid. This 3-phosphoglyceric acid finally form the C3 cycle. So finally form the C3 cycle. It is phosphorylated. So phosphorylation means ATP is utilized and ADP is formed. So clear. This is the another cycle is called photorespiration. So what happened? What are the main reactions in photorespiration? So in photorespiration, high amount of oxygen and low amount of carbon dioxide, the RUBP react with oxygen in presence of enzyme RUBP oxygenase to form 2-phosphoglycolic acid and 3-phosphoglyceric acid. This 2-phosphoglycolic acid dephosphorylated to form glycolate. This glycolate migrates into the peroxisomes. In peroxisomes, it is oxidized to form glyoxylate and hydrogen peroxide. This hydrogen peroxide, in presence of enzyme catalase, it split to form water and oxygen. So this is the important reaction. In photorespiration, the hydrogen peroxide. In photorespiration, the hydrogen peroxide is formed in peroxisomes. In photorespiration, the hydrogen peroxide is formed in peroxisomes. And next one, the glyoxylate is converted into glycine. This glycine is transferred to mitochondria. In mitochondria, it is decarboxylated to form serine. This reaction is catalyzed by the enzyme serine hydroxymethyl transferase. This serine again transferred to peroxisomes. In peroxisomes, it is transaminated to form hydroxypyruvate. Hydroxypyruvate is reduced in presence of enzyme hydroxypyruvate reductase to form glyceric acid. The glyceric acid formed in peroxisomes transferred to chloroplast. In chloroplast, the glyceric acid is phosphorylated in presence of enzyme kinase. So, in presence of enzyme kinase to form 3-phosphoglyceric acid. As a result, final product is 3-phosphoglyceric acid. So it is enters into the C3 cycle. So this is the another cycle. It is called as photorespiration or C2 cycle. So what is the importance of C2 cycle or photorespiration? సో దీనికి కనుక తీసుకుంటే సో ఇంపార్టెన్స్ కనుక తీసుకుంటే సో ఇట్ హ్యాస్ నో పాజిటివ్ ఫంక్షన్. సో ఇట్ హ్యాస్ నో పాజిటివ్ ఫంక్షన్. So next one, this photorespiration is inhibited by, this is the important one. The photorespiration is inhibited by a chemical substance. It is called alpha hydroxy sulfonates. Alpha hydroxy sulfonates inhibit the photorespiration. The photorespiration is inhibited by a chemical substance. It is called alpha hydroxy sulfonates. Inhibit the photorespiration. The photorespiration is completely depends on C3 cycle. It is completely depends on C3 cycle. So clear. This is the another cycle. It is called C2 cycle. Photorespiration or C2 cycle.
సార్ హలో సార్ హలో హలో
చెప్పరా.
సార్ ఒకసారి సెషన్ ఎండ్ చేయండి, మళ్ళీ కొత్త టాపిక్ అప్పుడు మళ్ళీ కొత్తగా స్టార్ట్ చేయాలి.
ఓకే, ఇప్పుడు దీన్ని ఎగ్జిట్ చేసి మళ్ళీ లోపలికి వస్తా.
ఆఫ్టర్నూన్